Okay, hold on one
This
second.
meeting is being recorded.
Okay. All right. Hold on. You can edit this out in post-production. Okay. All right. So welcome, everybody. I am super thrilled to introduce our speaker and guests today from the laboratory of Manolis Kellis, who is probably well known to many people because he is a world leader in computational genomics and computer science and all kinds of basic biology, transcriptomics, et cetera, too numerous to count. But he's also been active and interested in his lab in a variety of things that intersect with some of our other interests, because we're also interested in that genomics and omics stuff. And that is leveraging, high dimensional data, risk prediction, EHR data, and so on. And so I happened upon this paper that our speaker, Ben, is it Lengerich or Lengerich?
Both works.
What do you like? Both work.
If you're authentic German, it's Lengerich, but I'm not, so Lengerich is fine.
I say. Okay. All right. We'll go with the Americanization or something. Anyway, so Ben's paper from this lab that, first of all, has an awesome title. You can't miss it: Death by Round Numbers and Sharp Thresholds, which I think is brilliant. But addresses methodological issues that we run into and that are so crucial for precision medicine, et cetera. And I just thought it would be fabulous to have them and have Ben talk about the research and impacts of the way the EHR real world data that we use so much is influenced by or lacks causal inference ability. So I'm thrilled that you've joined us, and Ben is in the introduction to the meeting today. You'll note is a postdoc in the MIT Computational Biology Lab with Manolis Kellis at MIT and CSAIL and at the Broad, and has been working on machine learning for, I guess, in a variety of contexts. So super excited to have you here, and hope it'll be the beginning of maybe collaborations as well. So
thank you so much. Thank you.
Turn it over. Yeah.
Reminder to switch the camera, and I'll give a brief introduction to Ben as well. So basically, thank you, Jordan, for the very, very kind words. It's been such a pleasure to interact with your group through many years now, and to also have dinner with you and all of these sort of chance encounters in this joint community that we have. And I just wanted to sort of briefly say that the work that you're going to hear today is sort of parallel to a lot of the work that we're doing in our lab, which is all about genetics and variation and patient heterogeneity. And what Ben has been working on is spanning many different areas. So Ben did his PhD in machine learning with Eric Xing, who's a world-renowned leader in machine learning at CMU, and who just recently started a whole new institute at the interface of machine learning and health and all of that. Ben has been working on EHR data for a long time now. He's been collaborating with several hospitals, and one of our hopes in having this conversation is that engaging Ben in sort of the MGH cohort and sort of the joint biobank that you have there, I think would be immensely fruitful, for both us and hopefully for you as well. And also that Ben works on many additional areas of machine learning. He's been looking at contextualized learning, where you can learn patient-specific networks by sort of learning across thousands of individuals simultaneously, but understanding the hierarchical structure of the groups that they form and the individual level groups at multiple levels of hierarchy. So this is only one of many different talents that he has. And at some point, he'll be in the job market soon. So hopefully, maybe he'll be one of your colleagues if you guys are hiring this year or next year. All right, so Ben, take it away.
All right, well, thank you, Manolis, and thank you, Jordan. I'll go ahead and share my screen here. Everybody can see that now? Good, good. Yeah, so like Manol's was saying, we've done a lot of work on high dimensional inference, all these complicated estimators of genomics, of EHRs, of real world effects. And in this paper, what we're really interested in is, what happens when we apply these to real world data? What do we actually learn from the data? What do they look like? If we have really good machine learning models, do they reveal things about the data that might have always been lurking there, but we never really realized before? And so we call this death by round numbers and sharp thresholds, because as we'll see, one of the major findings is that there are big influences from protocols, from thresholds that we actually pick up in the statistics. Yeah, so as we're all aware, reality doesn't always meet our expectations. Let's take a motivating example of mortality as a function of serum creatinine. So serum creatinine can indicate kidney failure. So we might expect that at higher levels of serum creatinine, patients in renal failure have higher likelihood of mortality. And maybe the curve looks linear, maybe there's some saturating effect, but we would generally expect for higher creatinine levels, higher mortality. In truth, that's actually not what we find when we apply this to real world EHR data. The real risk curve looks nothing like our idealized expected risk curve. And it's not just a little bit off, it's actually the trends are in the opposite direction of what we might expect. But if we look at this curve a little bit closer, we might start to think about why this is happening. There's this really suspicious change in slope at a round number of 3 mg per deciliter. So is this saying that there are changes in mortality risk that are triggered by round number thresholds? And similarly, there's a huge change at 5 mg. So it looks like these impacts of round number thresholds are actually having major impacts on the mortality rate. And we pick this up in our machine learning. And then finally, just overall, this is exactly the opposite of what we would expect. We would expect elevated creatinine to be linked with mortality, but actually it's the opposite. We get the opposite of what we would like to get. And this wasn't just one example. We didn't go looking for this. So I'll show you one data set here. This is a data set of pneumonia patients. It's a little bit of an old data set collected in 1989, but it has 46 features about the patients. So all of these risk curves that we're seeing are corrected for all other risk factors. It's not just a single impact, it's that impact after correcting for everything else. So in this one data set of pneumonia patients, we first find blood urea nitrogen has this interesting curve. And what's very interesting about it is that it's flat for a while and then jumps and then flattens again at about 35 mg per deciliter. If we break this curve down for men and for women, we get even more interesting findings. So for men, the risk decreases for very elevated BUN, but for women, it continues climbing. Maybe there's a difference in treatment at 100. If we look at heart rate, it looks like really fast heart rate. A racing heart, tachycardia, is good for you because the mortality rate, it rises in the middle and then it peaks at a moderate tachycardia. But then for very severe tachycardia, it doesn't get any worse and it actually gets mortality risk decreases. So is a racing heart rate good for you? And then finally, that previous example of the creatinine, that was actually from this data set. And the point is that the true low risk should be a low creatinine, but what we observe is that the best regime is the highest creatinine. So a blind AI model might look at this and say, let's put everybody into renal failure, and then they'll get good treatment. They'll get really specific attention, and they'll all survive. Finally, we can also look at comorbidities, and these are also often giving us the exact wrong sign. So history of chest pain looks like it's really good for you. History of asthma looks like it's really good for you. History of chronic lung disease also looks like it's good for you. And we can imagine why are these effects happening? And chest pain, asthma, chronic lung disease, these are all things that patients know they have, and they're very obvious. And when you're being hospitalized for pneumonia, it takes an action on the patient's behalf to actually get hospitalized. So we can imagine that these patients that know they have some comorbidities with their cardiovascular system, they're showing up to the hospital more quickly, demanding better treatment, and it's actually reflecting in the mortality rates. So these effects are everywhere. This is one data set. We found maybe five types of effects here. Multiple comorbidities are going the wrong way. What are we going to do about this? And why is this happening? Well, this happens because, as we all know, our real world evidence is actually, it's not pure. It's coming from complex human behaviors. So we observe some biomarkers and we observe some patient outcomes. And we might want to learn a model that predicts outcome from biomarker. And then we can say something about the mechanism of the outcomes or potential therapeutic targets. But of course, it's not that simple. There's also, there's actually an underlying risk that we don't observe. And there are treatments that we may or may not observe. And here we're going to say treatment as a very broadly defined term. So that includes not just treatments that patients are given, but also the actions that are taken, their own understanding of their disease, their priority, all sorts of things. But we're just going to use treatment as a very broad word. So we may or may not observe some treatments. We probably won't observe all treatments. And we will never know if we have observed all treatments. And so as a result, it's not clear if our model that predicts outcome from biomarker, if it actually tells us very much about the underlying risk. And this gets a very complicated feedback. So the risk, the underlying risk makes these biomarkers, and the biomarkers affect treatment decisions. The treatment decisions can improve patient outcomes, but the risk works against that, worsening patient outcomes. So we have these two pathways that are competing against one another. As you get more sick, you might get more treatment, but you're more likely to have bad outcomes. On the other hand, if you get less sick, less apparently sick, then you may be less likely to get treatment, but you were healthier in the first place. So this is a really hard problem for data-driven real-world evidence.
Before you move on, I want to spend just a teeny bit more on this slide because it's just so complex and so important. Basically, the question is, if I see a biomarker, is it increasing or decreasing patient outcome? And the traditional view that everybody has is the upstairs one, basically that if I observe the biomarker, that basically tells me something about the risk. So it brings information to the risk. And that risk basically tells me that the patient is going to fare worse. So we basically have a positive correlation here and a positive correlation here. The true causality comes from the risk. Basically, you start here, and then the true causality flows this way and that way. And what the biomarker is telling you is that your initial risk was higher. So I think, the naive models basically focus on the top. And I think what we are trying to elucidate in this paper and what all these curves are doing are basically saying that when you have a biomarker, of course, the decision at the clinical level is to recognize that you have a higher risk. So basically the prediction flows that way, even though the causality flows that way. And then to intervene and get a treatment. So basically you have on one hand, the increased risk coming this way, and that's the positive red arrow where you're predicting a worse outcome. And then, but what's happening here is that what we're observing in all these counterfactual, all these sort of inverse correlations, where the higher the biomarker, the lower the risk, is this competing path between the two. So basically you end up with this jagged curve where every intervention kind of brings you back. And at some point you're getting the best treatment possible. And this ends up being actually lower than the starting point. So that, it's these two competing models that we're trying to elucidate here. I hope everybody's with us. Go ahead, Ben. Are there any questions, first of all? Because the whole point is for it to be interactive. So if there's any questions, we can actually see you guys. You can just raise your hand and say it, and Chancellor will hear you.
Can you hear me?
Yes, we can. In fact, we can hear you twice. So
Can you hear me now?
Very well.
Make sure that I'm muted here. Okay.
Perfect.
Yeah, this is really as fascinating. I wonder if you don't see the, first of all, I'm thinking about in the case of many of the things that we look at in psychiatry, we probably don't have this problem as much because we don't have great biomarkers that we know are actually targeting the biology. But let's say that we do. Biomarkers can be either on the causal pathway or they can be kind of either consequences of the disease, or they can be sort of correlates, essentially. And if you don't see this effect, in some ways, that might be informative about whether this biomarker is actually targeted by the biology, whether your treatment is targeting the biology.
I love it. So your intuition is so elegant. It basically says that, well, every time you're using a biomarker to decide upon treatment, you should be seeing these thresholds. And if a biomarker is actually being used, but all you see is this, it basically means that the biomarker is not functioning. Right?
Right. I'm trying to think of, like, for example, let's say it was LDL.
Yeah.
And you're using statins. So maybe you would see this, you would expect to see this.
Yeah.
But let's say you're using, it's LDL and you're actually looking at the effect of, I don't know, coronary bypass surgery or something, or, stenting.
Exactly.
Where it's not actually affecting that pathway, it's mechanically affecting risk.
So I think what's unique about Ben's work is that he doesn't actually know what the treatment is, at least in the first side, basically. So it doesn't, basically, if there's any treatment that's triggered by that, or by anything else that correlates with that, then we'll see it. But it doesn't matter which treatment, as long as there's some treatment. So basically, if they do coronary bypass based on this and it's not helping, then indeed you'll see a straight curve, and that basically means that we're using the wrong biomarker. Or no, it means that the surgery is not helping, basically.
Amy.
The other thing to consider here, on top of what Jordan pointed out, is this a death by round numbers, or is this compressing all time points into a single cross section? And so, yes, your creatinine should look protective when you're at 7 or 8 because you're on dialysis, right? But folks who don't elect to go on dialysis die when their creatinine rises above, say, 5-ish. Or people come to the hospital when their creatinine rises above 3.5. If you're at the hospital for a creatinine lower than that, then the reason you're at the hospital in a real world sense is for a totally different reason. So the point here is not that there aren't round numbers. The point is that you are flattening them into a single observation, which can also cause some of your issues. And depending on what models you use, repeated measures are also going to be challenging there.
Exactly. So, and one of our points, one of our perspectives is that this is kind of a fundamental problem. Like what you're saying is that if we use our intuition of how the medical system works, we know how people get treated for creatinine, we know when they come to the hospital. We can think about experiments, how to split the data that would fix this. But we may or may not know that before we actually run the model, before we look at the EHR data. We might not realize these things are lurking in there.
Well, you might not need to know in advance if what you had was, if time was one of your axes. That's what I'm actually saying.
So there are some things that time would fix, but I don't think the time would fix all ails. Right.
There's two thoughts that come to mind. The first one is the little drawing that I'm playing here with all the colors of Zoom, which is kind of cute. Sorry, this is the 5-year-old in me resurfacing all the time. But basically, the image that comes to mind when Ben is showing this sort of opposite effect going down, when in fact each of the individual curves goes up, is in other places of statistics known as Simpson's paradox. The fact that in every individual subgroup, the trend is up, but if you look across subgroups, the trend is down. And I think at some point, Ben, you should sort of look at Simpson's paradox and how, we can sort of loop that into the paper. So that's the first thought. The second thought is the whole death by round numbers part. So basically, at some point, you're thinking, when do I intervene? Basically, if you want the curve to basically be flattened out, what you would like is the risk to sort of stay stable. And that basically means that at every threshold, whether it's round or not, I'm basically providing some kind of incremental therapy, which, maybe when we all have wearables and, they can dispense drugs in our veins as soon as sort of something rises or falls. As soon as I get angry, somehow, the right neurotransmitters will be released in the back of my neck and I'll feel calm again. So then you'll end up seeing all these sort of flat curves. But that's at the individual level. And that's where, ergodic systems basically come in, where you basically say, this is at the patient level, but at the population level, the other question is, how do you flatten that curve? Well, at the patient level, by releasing a tiny little amount of the therapy, which of course is not going to be feasible. At the population level, it's by flipping a coin and basically saying, I'm going to flip a coin for each patient. And when they're in this regime here, I will have two chances in 10 of admitting them, and here 3 chances of 10, and here 4, and here 5, and here 100%, etc. Where you're sort of flattening things out in the sort of risk curve at the population level. The reason why I'm saying all this is because your question was, what's contributing to the death? Is it that we're not doing the right things by doing these interventions? Is it that we should be intervening earlier, but then there's a cost benefit analysis? Who do you give this to? Maybe that's where the flipping the coin goes in across the healthcare system, but then, nobody wants to be at the side of the coin where they don't get treated. And maybe some of that is also looking at additional biomarkers. So basically here, we're looking at one variable at a time, but basically in an EHR setting where you have multiple variables, you might be able to flatten these curves by sort of recognizing who's at higher risk or lower risk, whether there's, slightly past or slightly below the threshold, by looking at additional variables that might be informative. Anyway, just some thoughts for addressing.
Yeah. I mean, in some ways, isn't this the familiar problem of confounding by indication, where methods for that include, and I don't know that they work all that well, but like propensity score matching that, attempts to essentially even out the underlying risks between people treated and not treated, right? How do you, or are you going to mention that, or how does that fit in here?
No, that's exactly the right intuition, is to say, if we were able to observe all of these treatments or the propensity for the treatments, we could correct for them and then estimate the true underlying risk. But our perspective is that that's a goal. It's a platonic ideal. But we're never really going to achieve that. So we need some methods that are able to look at the risk curves that we've estimated, and then allow us to use our medical knowledge to say, do these risk curves look right? Do they look like there's something else that we're picking up that should be corrected later?
There's another thing that I think is related, which I put in the chat, which is basically known as, sorry, let me just get the name right. Goodhart's law, which basically is related to test taking. For example, when standardized tests became a norm, they were a very good metric and a measure of success of a school, but then suddenly they became the target. And they ceased being a good measure because they were suddenly a target. In other words, now students are studying for the test. So by changing the test, maybe the best way to do well at the test by constantly changing the test is to simply just learn the material. But if the test becomes standardized, then, cheating becomes basically teaching to the test becomes easier. And at some point, you just if you keep changing that metric, you kind of give up. And the easiest way to sort of do well at the test is to actually learn the material. And that's sort of the concept here. Basically, if it's a measure of risk that becomes the target for treatment or the decision point for treatment, then it stops being a good measure for risk. Anyway, I think that's related to the whole thing.
Yeah.
Okay. Any other comments, questions? If not, Ben, maybe take it away.
Very good. So along these lines, our perspective is that we might be able to work backwards to estimate this underlying risk. Instead of saying, let's perfectly specify all the treatments that influence this first.
I apologize. Before you go on, I wanted to make one more thing, which is the reason why this matters is not because doctors don't know this. Doctors are very smart and they know this. The problem is that AI and machine learning models are very dumb. They don't have that natural intuition. So basically, when, as we're moving towards more intervention of AI models that will then learn all kinds of things from the world and start making predictions about how to intervene, maybe not in a medical setting, but, in an airline safety setting, or in a sort of, nuclear plant safety setting, et cetera. These kinds of sort of interventional curves that are sort of shifting the trend can basically lead to very dangerous recommendations. And as we move to much more immersion of these, large AI models, we want to basically make systematic changes that can allow these models to not be confused by these types of things. So that's sort of one of our fundamental goals here. Like, doctors, of course, know this, but AI systems don't.
Right. And this is very near and dear to our hearts, because if we get funding, which looks like it might happen, we're going to actually deliver suicide risk prediction, AI-driven suicide risk prediction models into the emergency room. And we'll be looking at, the impact on those outcomes. And you certainly want to avoid this situation where it starts to, especially if you're doing, I guess, reinforcement learning kinds of things, right? Because you're sort of modifying the thing along.
Yeah.
Oh, sorry. Can you hear me?
Yeah, of course, very well.
Yeah. Oh, okay. Yeah, I was saying, especially if you're maybe doing a reinforcement kind of learning where you're modifying things as the picture changes, that could be problematic. I'm just in the middle of reading that book, which I think is great, and you've probably read, The Alignment Problem. Have you read that book?
Not yet.
It's awesome. Really good. Anyway, it's about all the ways that AI and machine learning can go wrong, basically.
The other.
Go ahead.
I'm sorry, I cannot see you, so I don't know when you're about to stop. So that's why I keep interrupting you. I apologize. So the other thing that comes to mind, for example, in the very specific case of suicide, you could say that a suicide note could trigger an intervention, which will then reduce the risk. So you don't want the system to learn that sort of when the person posts on Facebook that they're about to suicide, that suddenly they're not at risk anymore because, they're fine. Because it goes the other way around.
Right.
Okay.
All right.
Continue, Ben. Sorry.
Yeah, thank you. That's an amazing motivating example too. So our perspective is that we might be able to work backwards to estimate this underlying risk. We only get to observe treatment protocols, real-world treatment protocols, based on distributions of treatments, and we get to observe the actual risk that happened. But that risk is a factor of all these combinations. So the question is, how do we estimate this underlying risk just observing the treatment protocols and the population risk? And if you'll notice in this diagram, we have two different thresholds for this treatment, which changes the risk curve from a red to a blue. And in this case, we can imagine that if the treatment protocol is black, it results in this shape risk curve. If the treatment protocol is the higher threshold, the yellow, it results in this more strange looking risk curve. So the risk goes up, it peaks, and then it hits the threshold, and then it comes back down. So the difference between these thresholds is how much the risk is aligned with the difference in risk based on the treatments. If it's misaligned, then we get these weird shapes in the graph that alert us to the fact that we might have misaligned thresholds and the proverbial death by round numbers. So our goal is, can we look for these shapes so that we can use, doctors' intuition? They know how the system works. How can we combine AI systems that don't know how the system works with doctors that know how it works? And our solution is to say, if we express things in these shapes, we can use medical knowledge with the data-driven evidence. This takes some machine learning to do. The way to estimate these is using glass box models. Glass box means interpretable. We can look at it and it's exactly what the model estimated. It's not some approximation, it's exact. And the way we do this is using a glass box model type called the generalized additive model. So generalized additive model, for anybody that's not familiar, says that individual variables each get their own function that changes the outcome. And what this allows us a glass box inspection because we can look at the impact of each variable on its own. The model estimation does its best to get rid of correlation. Of course, we need to keep this in mind when we look at it, but the model has already done its best to get rid of the correlated effects. And what this looks like is that we can express some complicated outcome as a sum of individual variable graphs. And the graph is the function. So if at some point we wanted to modify the graph to express a different function, we could do that. If we wanted to look at what the graph learned in a particular region, we could do that too. So coming back to our Grandin example, we might have this complicated shape, and we might be able to break it down into a couple of different components where we have more interpretable shapes that correspond to the thresholds of interventions. And historically, additive models may have sacrificed accuracy for this interpretability. So they said we're going to have separable components, and that's going to make it interpretable, but they're not going to be that accurate. One example of this is linear or logistic regression. These are additive models, but they're not very accurate. They can't model nonlinear effects. They can't model the very common healthcare case of where there's some marker and the middle regime is healthy, but both extremes are unhealthy. Linear logistic regression can't handle this unless we've already pre-specified those bins. And because of this, it often gets the sign wrong. It says there's a negative effect just because there's two regimes on either side. So just graphically, in the same decomposition, linear logistic regression might look like flat lines, but that's not actually going to tell us what we want to learn from this graph. We want to learn there's some reasonable biological signal, but it's been perturbed by our interventions. But really, additive models don't need to be linear components. Are you able to hear me still? Oh, we're changing the camera. Okay, good. So they don't need to be linear components. We can actually make these complicated while keeping interpretability because we have the separability already. So we've explored two ways of putting additive models on modern machine learning steroids. And our solution to this is to say each one of these components can individually be very, very complicated. But together, we're still going to have the separability so we can inspect each component on its own. The first way we did this was using deep learning. So we said each component can be a deep neural network. And this gives us the power of deep learning, but without sacrificing interpretability. And the second way we did this was by making each component be a boosted tree. And boosted trees use cuts in the data to split up domains, split up features, and make individual predictions for each cut. And we actually find that the boosted tree method does better than the deep learning method in healthcare precisely because of this round number effect. There are thresholds that change outcomes in discontinuous ways, and the deep learning likes to smooth out discontinuities, whereas the trees like to split the discontinuities. So we actually—
Just to clarify, it's because of the sharp threshold, not the round numbers.
Yes. It's a sharp threshold, which often happens at round numbers.
Exactly.
Yeah. So these trees are really good for recovering them because they're looking for places to split. So they will report to us where it's split. We call this an explainable boosting machine, an EBM. And what they do is they return these jumpy but interpretable components. And just to reiterate, just like logistic regression, our final risk score is the sum of all these individual risks. And we get these graphs that are interpretable for each feature. We get some score associated with it for the patient, and then we sum those up, and that gives us the overall patient risk score. If these were logistic regression, it would just look like flat lines. So as a result, for most healthcare data, most tabular healthcare data, these EBMs are actually state of the art. We don't really give up accuracy on our tabular data sets. We've done some benchmarking and we find this is state of the art. It's also better than things like XGBoost, which might have more complexity. So given these high resolution models, they allow us to zoom in very tightly into the risk curve. And that's what we're looking for, to see these discontinuities, these strange patterns that might tell us about underlying treatment effects. So this gives us both the high resolution and the interpretability, and allows us to say, if we see these strange looking risk curves, they might actually suggest that there are treatment protocols that are misaligned with the statistically optimal threshold. In this example, the top one has this green threshold that's too low, where the black is statistically optimal. And as a result, there's excess risk, where the green curve goes up rather sharply and then plateaus, whereas the black curve is a little more smooth and then a plateau. And then we saw this yellow line before, where the threshold for treatment was too high, and as a result, the risk curve goes up and then turns around, comes back down in a pretty sharp way. I see a question. I think I see Jordan has a question.
I think he's walking up to the mic.
Okay, perfect.
Okay, can you hear me now?
Yeah,
Yes.
he's going behind the camera to ask the question.
Yeah. Yeah, I love this. This is so cool. So two questions. One is, so what you're saying is this is not only a method to dissect and sort of assay for these paradoxical effects, but it actually will give you better but also still interpretable predictions. Right? That's
Exactly true.
And one question I have is, how do you tell apart this question of misalignment with the weakness or strength of the treatment, or does that matter? In other words, like if you have a treatment that's doing this, but it's not a very good effective treatment, maybe that doesn't matter, that you won't see the effect.
Correct.
You won't see these weird discontinuities, but maybe, I don't know what that means. Maybe that means it's not the greatest treatment, but at least it's not screwing up your predictions or something.
If the treatment actually makes the patient worse, then you'll see a very, very sharp discontinuity. Actually, you'll see discontinuity in the other way. And I don't think we see those, because I think, so what we've learned through this as well is that the healthcare system is doing a pretty good job at improving itself. And I think Ben will show some figures along that line. But basically, the hospitals are doing a pretty good job in sort of deciding that a treatment really sucks and not using it. So
well, so what if you were to apply this to a long-term clinical trial, randomized trial, and you've got a placebo, you would expect, would you expect, well, if it was a clinical trial, you'd be controlling the treatments too much.
Well, if it's a clinical trial where there's a point, then yes, basically, if there's a biomarker associated with a clinical trial that decides the intervention, then this model applies. If it's randomly choosing individuals, then it doesn't.
Right. And
Yeah,
what
actually,
is the, sorry, go ahead.
No, we just had a little bit of a timely example with that with the COVID, where in the beginning of COVID, physicians were semi-randomly assigning treatments. And then once papers began to be published on the effectiveness of things like glucocorticoids, we start seeing these effects.
Yeah, so in general, what we've seen is that the healthcare system adjusts quite rapidly because doctors see what's happening.
Yeah.
Right. Right. And these are, and that presumably it's biomarker, like a genetic test or a pharmacogenetic test wouldn't be susceptible to this, right? Like if you were looking at P450 enzyme variation and stratifying people for different antidepressants like we do.
I think it would, because it basically, the biomarker itself can be indicative of what other factors are contributing. And as you're sort of using the biomarker as an intervention, then you'll see this discontinuity along the biomarker curve.
Oh, yeah. Okay. Yep.
Yeah, there's this,
Cool. All right.
there's kind of this interesting regime where if you do nothing, you see the underlying risk curve. And if you have a perfect treatment, perfect system, it's, it's flat, but it's somewhere in the middle that you're making informed decisions and you're doing something, but you're not doing everything. And then you get a strange risk curve.
Right. Excellent. Okay.
So just to rephrase Goodhart's law, basically when a risk measure guides intervention, it ceases to be a good risk measure.
Right.
But basically, if it doesn't guide intervention, then you don't have to worry about it. And if the intervention has no effect, then it's not an intervention. So again, that discontinuity will not appear.
Got it.
Okay, carry on. Ben. I'm wary of time as well. The discussion is the best part, but we should still go.
So we group these into two classes of treatment effects. The first is these discontinuities, that there's some sharp threshold, so the risk changes dramatically at this threshold. We've looked at this before, the BUN, when it gets to 35 or the 40 region, it jumps up, but then it's flat. And similarly, at 100 for males and females, it does different things. Also, we see this in patient ages, where this isn't something you're going to modify, but it guides interventions. When we think about patients in particular ways, these seem to change in social biases, round numbers of ages. We can see discontinuous effects. It's kind of also hinted at in this graph. It's a little blurry, but actually we see in this old pneumonia data set that for men at 100, mortality rate jumps up, as might be expected, but for women at 100, mortality rate goes down. And there's, there may be explanations for that. Maybe the old phrase of pneumonia being old man's best friend could be one explanation. So the second type of treatment effect or countercausal effect, we call these countercausal low risk regimes. So these are smoother varying effects, but because aggressive treatment is correlated with risky biomarkers, we actually see an entire regime that looks wrong. So it's a low risk, but it's actually inherently high risk. So these are all the comorbidities. We say that we could, for Boolean comorbidities where it's you either have it or you don't, we could think about these as discontinuous, but all of them are discontinuous. It's really the countercausal ones that we're interested in. And then there's also the tachycardia example, where once you're above this tachycardia, you're getting some sort of good treatment. And again, for the creatinine, you're probably on CRRT or dialysis or something like that. So those examples were relatively strong because they all came from this 1989 example. But as Manolis has been hinting, the healthcare system has gotten better over decades. And we're able to see this by looking at the MIMIC data set. You might be familiar with MIMIC. It's a very widely used EHR data set from Beth Israel. There's tens of thousands of hospitalizations and 3 iterations of it. We select patients admitted to the ICU here, and we look at mortality. We try to predict mortality for them. And in the final version of the MIMIC, we get the treatments, so we can link treatments to these effects. So first, we can look at the same risk curve of creatinine. And again, we actually see a similar risk curve as we saw in the pneumonia data set. So for these very elevated creatinine patients, they're getting aggressive dialysis CRRT, and it's working. As a result, the lowest risk patients are those really high CRRT, maybe renal failure. Excuse me. And what that means, that matters because an AI system that just naively learns this risk might try to push everybody into this regime. So thinking about, of course, that suicide prevention example, we could imagine something that predicts good outcomes because of intervention, and that people are encouraged to go into that regime. That would be disastrous. Given the treatments in MIMIC-4, we can link these risk curves to the actual treatments. And we do see the dialysis and the CRRT are making these decisions at the round numbers of the 3 and the 6 mg per deciliter. And that agrees with a lot of these changes where the risk reverses direction, where it jumps down as the treatment changes.
Sorry, one other question there. This goes back to, I think, what Amy was asking. So when are these people are having multiple creatinines measured. When are you using the creatinine for this? Or which creatinines are you using?
Yeah, so this is on admission for this.
On admission?
Yeah.
Okay, got it.
because we're imagining the scenario where, right, somebody comes in and you have an AI system that's built on it. So you could imagine, building a time varying thing with a time varying model with interventions baked in, and we estimate the effect of the time varying. But that wouldn't really change the fundamental picture, which is there are these interventions that are being informed by the risk, and we want to know whether or not the interventions are changing risk in these strange ways. So another example is this sodium, is serum sodium, which can indicate dehydration or other processes. And we see the classic healthcare shape where there's some healthy region surrounded by higher risk and high risk on both sides, but it's not exactly what we would expect. And it changes over the different data sets. In particular, in the most recent data set, the black one, we see that risk rises rapidly from 140 to 150, but then it stops and reverses direction. And this, of course, is a nice round number of 150, in which we can see changes in oral intake, changes in electrolyte replacement, prompted or apparently prompted by some round number effects. As a result, these patients, an AI system might learn it's better to be really dehydrated than be moderately dehydrated because you might get more attention then. Also, patient age has these effects. We can't modify patient age, but it can still inform our care. And if we zoom in, we can see round number effects at a bunch of different ages. And they actually, we could imagine they change over time as social impacts also change, as retirement age used to be more uniform and now it's less uniform. Some of these effects have gotten smaller. One very interesting effect is that 80 years old, the choice of catheterization switches. So this changes from an ileal conduit, which requires an incision, to a condom catheter, which is not. So there's an evaluation of exact measurements of input-output versus infection risk versus modulation. And when patients hit 80, there's a step function decrease in the risk, or in the decisions. Similarly, this is Dilaudid, a pain medication. At 50, we see a step function decline in pain medication prescriptions. Looking at BUN again, blood urea nitrogen, we can see over these 2 decades, hospitals have gotten much better at treating patients with elevated BUN. The outcomes are much better, and this drives us to the question of, what do we want these risk curves to look like at the end of the day once we've made perfect medicine? Should they be completely flat? Should they look exactly like an underlying risk curve? Or should they have the underlying risk curve but modulated down with proportional benefits for everyone? I'm not entirely sure of the right answer there, but I think they should be flat, or they should be smooth at least, because these discontinuities suggest that we're doing better for one group of patients, but we could do just as well for a nearby group of patients. So it's encouraging that the impact of the elevated BUN has gotten better over years. But we still see, even in the most recent data set, we see some of these strange effects. So the risk goes up, 40 comes back down, and then is mostly flat, and then there's a decrease at 80. So at 40, we see a jump in the CRRT usage again. So again, this seems to be suggesting that CRRT is very effective at preventing this in-hospital mortality, but it's rationed in some way. And I think there have been ongoing debates about whether or not CRRT should be expanded to more patients. Maybe this suggests that it could be used at lower thresholds. And then finally, there are more treatments being given for extremely elevated BUN. In this example, there's not as strong indications of what this sharp decrease at 80 BUN would be. We don't have great treatments that are linking to that, but we just see a general increase of treatments, of the number of treatments when they cross this threshold. So what are the implications of this sort of paradigm? We know confounding exists, but what do we do when we know that confounding is pervasive, it's ubiquitous, and it's so strong that it gets us the opposite signal from many data-driven analyses, even when these analyses predict very accurate outcomes? So for clinicians, we might like to think about our thresholds very specifically because they matter. We can see if we have a threshold that is biased to a round number, it might be missing patients that are right below or right above that round number. There's also an idea of a subclinical abnormality. So we might want to study these states of subclinical changes where we haven't quite reached the threshold to prompt treatment. But actual clinicians know to be looking out for this, and they might change behavior. It might be worth doing more studies to say, how much does the subclinical change? How much does it change the outcomes? And then finally, this is also getting back to the questions from Jordan and Amy. What would the end goal look like? If we have, are we always going to be threshold-based medicine? And in some case, yes, because as Manolis was saying, we're not going to want to flip a coin for each individual patient, although that would smooth out the risk curve. We don't want to flip a coin for them. So how do we get a smooth risk curve without resorting to randomness? And the answer is to align the underlying risk with the decisions perfectly. So the reason we have these changes is because the threshold is based on one axis of variation, but not a continuously varying dose or a holistic picture of risk. So if we can combine all these holistic evaluations, multiple time points, we can make better decisions, we can make better thresholds, and we can make the threshold align to the actual risk. And then also for machine learning statisticians, people applying these methods to EHR data sets, it's important to not just blindly apply ML models to healthcare. All these models that I've shown are very accurate. They're state-of-the-art at predicting outcomes on these data sets. And the MIMIC data set is used by thousands of people. But there are these very strong effects in it that get us the opposite implications of a lot of these biomarkers. And all accurate models are going to have learned these treatment effects, but they might not tell us that they learned them. And if we're just relying on accuracy as a measure for how good our model is, we're going to be missing out on a lot. And if we go and deploy that, it could be doing exactly the opposite thing that it should be doing, just gaming the healthcare system instead of actually fixing the underlying problem.
And that's something that's pervasive, where basically increased accuracy very often leads to increased bias. And that's something that I think, we need to cope with as a field. The fact that increasing the power of your model will only make it more inaccurate or more biased in a particular direction. So it's not a question of power, it's a question of bias, and you cannot, escape that without sort of stepping back and realizing that these are, biased by these treatment effects.
Yeah, that's exactly right. So once we can recapitulate the real world, what should we do? Yeah, very good. So I guess we're close on time, but I'll just close with a recent personal story. Last week, I was on vacation with my wife, actually, in Acadia National Park, where it's not great self-service, but it's nice for a vacation. And we went up to the top of the mountain there, Cadillac Mountain, and we got some cell servers up there. So after we took this picture, we got a phone call, and the phone call said, actually, our family member is in the hospital for some heart problems. And this is a family member that's very healthy, shouldn't really be having heart problems, unexpected. And so my wife, actually, coincidentally, is a nurse in a cardiac unit. So she was very interested to get all of the labs, all of the values for this family member to try to understand what's going on. And after a while, after a couple of hours of driving around Acadia trying to find better cell service, she eventually asked for a magnesium lab. Magnesium is, of course, an ion that can change the electrochemical signals in the heart and in other places. And so it's important to be in balance for that. And what we got back was a reading that was below a threshold at my wife's workplace, at her cardiac ICU, but it was above the threshold for treatment at this community hospital where the family member was being treated. And my wife says, you know, let's start treating this. You're below the threshold that I care about, that I've been trained to care about. And the community hospital said, well, you're at 1.8 and we only really care about 1.7. So you're probably fine. Eventually, they did decide to treat it. And the family member got better. When they went home, we found out that they might have some underlying issues with electrolyte and ion balances. They've had this issue before, actually, in the past, once we knew to look for it. So this was an example of where we had this maybe a different threshold combining with treatments to produce possibly some excess risk right above the threshold of treatment.
I think this example illustrates so many of your points. The first is it can happen to you. This is not just like some theoretical paper. This is like a very real story. This actually happened last week to the author of the paper. The second is that the variation in threshold is something that you might think that what's causing these changes, but in fact there might actually be stochasticity and sort of differences within the same data set, because some doctors might choose to take different thresholds. The third one is gosh, I forgot the third one. Never mind. Yeah, the third was the most important, and I'll get to it as soon as you.
I think the third one is try not to go to a community hospital, maybe.
Yeah.
That's really, yeah, no, you're right. I mean, that sort of brings it home, and it's really fascinating. I wonder.
Sorry. What was the point that Ben made about the holistic approach? The fact that this particular patient, had we known about the excess risk coming to another, because of a precondition, would have been perhaps the threshold would have been adjusted even by the community hospital by basically saying, well, this patient is at risk, so 170 should be moved up as a threshold, and we're going to start treating earlier or something like that. So this is the holistic approach where all of these curves are based on a single variable, but with an integrative approach that looks at multiple variables, you can sort of perhaps better assess the risk and then flatten out these curves, not because of the threshold, which in a Bayesian framework, in any kind of naive Bayes classifier, with a single value, you can only choose a single threshold. But by sort of surrounding this with additional information, you might be able to make better choices. Sorry, that was the third point. Go ahead.
So we are, I guess we're out of time, but I'm going to ask one other question, which is, so in these generalized additive models where you're now estimating the each feature's effect using, say, boosted trees or deep learning, you're then using that for the prediction, not just for the diagnosis of the model's confounding effect, but also for a more accurate, smoothed prediction. Is that right? And I'm still not totally clear on how it. Yeah.
No, that's right. So one of the motivations of the additive models is to say that the interpretation of the model is the same as the model. So when we look at these things to diagnose the model or to diagnose the data, if we wanted to then deploy it, we could just draw a different line on the graph, and that would change the model. If we just wanted to get rid of that one effect, we could draw that. Of course, in deployment, it's always a question of should you trust your knowledge of what the model should have learned, or should you trust your data? Lots of complicated situation specific questions there.
Great.
Amy?
Amy, last question goes to you.
Last one, I'll be quick. Ben, this is really interesting, really great. A totally different approach than I've seen before. A core to the approach seems to be the idea that ultimately, after correcting for your treatment or intervention, that there is some true single relationship between a lab value and mortality. Do you think that is always true? And as you look across multiple data sets, maybe ones that have more diverse abilities to die, do you find that you struggle with that? Is it possible that there isn't a single relationship or a single curve between, say, creatinine and death or BUN and death, but one of a set fixed number that you ultimately want to adjust down to?
Yeah, you're tempting me so much, because as Manolis teased in the beginning of this this presentation, my other research interest is about contextualized machine learning, where we say that for different patients, there are different risk curves. And I totally agree with the premise of the question that these risk curves might be different for different individuals based on a holistic understanding of that patient, their background, all these sorts of things. And yeah, we're working on methods to estimate those things. It's just always a question of how do you interpret personalized curves versus how do we interpret population risk curves and population interventions.
Can I jump in a little bit as well here? So basically, Amy, there's another component. Basically, there's the interventional component of the healthcare system, but then there's the interventional component of someone's own body. So basically, as my creatinine levels rise and rise and rise and rise, at some point, there are other system failures that might be happening that might sort of make the risk increase even more rapidly. Or there might be feedback loops in my own body where suddenly, when you're suffering from hypothermia, at some point your body shuts off your extremities to sort of preserve, the core, etc. So basically, if you're just measuring core temperature, there are interventions from your own body that will basically sacrifice some organs in order to save others, etc. So basically, we have to think about both compounding effects due to systemic failures. You can't tolerate any more lipids in your blood after, or in your fat, fat cells become exhausted after 30 years of obesity. And then, there's compounding effects because now you can't store it anymore. So basically the risk that was kind of linear now becomes, hyperlinear. And then there's others where sort of there's feedback mechanisms that might reduce it. So, I mean, we see that with global warming, for example. There's only so much carbon that the ocean can store. And after a certain level, you basically have that failure of that system and failure of another system, et cetera. So I think this is pervasive, not just for healthcare systems, but for, systems more generally, where not just interventions externally, but also internal sort of feedback loops that can be positive or negative.
Yep. Okay, that was really great. And I hope we'll have a chance to continue this conversation and figure out ways to collaborate, because as I say, a lot of the stuff we're doing bears specifically on this, developing precision treatment rules and developing risk algorithms that we then want to intervene on, and we don't want disasters.
Thank you for
and we want and we want interpretable models, because clinicians are not going to accept,
exactly,
for many things, they're not going to accept models that are so black box that.
exactly.
All right. So thank you both. Thank you all.
Excited to collaborate.
Great talk, Ben. Yeah.
Thank
Beautiful. Have a wonderful weekend.
You too. Bye.
Talk soon, I hope.
Thank you, Ben.
All
Thanks,
right.
everyone.
Bye.
Thanks, Amy. Bye, everyone.